If you remember this slide, I was talking about the rate of incorporation of nucleotides if we perform the PCR reaction at room temperature, if we don't have ice available to us. But I never meant that you cannot perform the PCR on room temperature if you don't have a problem with non-specific amplifications and you perform a PCR reaction at room temperature, it's okay. But there are always chances that due to low incorporation rate of the nucleotides at ambient temperatures, there might be some problem with the non-specific amplification. So if you're getting non-specific amplification by doing PCR at room temperature, don't do it. Then shift your PCR reaction to 4 degrees. I mean on ice. Perform everything on ice. And you don't need to remember these figures, But you need to remember one thing, that the Taq polymerase or any other kind of polymerase, it keeps on incorporating nucleotides. So nucleotide incorporation starts whenever you complete a PCR reaction. Okay? So in between that, setting up the reaction, you are adding different things, and because all the things, they are not available. So the binding of the primer with the template can take place at very low temperature. But as soon as all the other ingredients, they are complete, and you leave your PCR mixture at room temperature for a while, there might be some incorporation of nucleotides, so you must take care of that. In case you don't have, if you don't have ice, then you can perform still the PCR at room temperature. So the next slide here is upon some of the problems that we can face during PCR amplification, and that is most oftenly two kind of problems. One is the non-specific amplification, okay, and the other is no amplification. If there is a non-specific amplification, definitely the answer is that your primer is binding non-specifically to the chromosomal or your template DNA at some position. It has got some homology there. And that's the only reason for getting non-specific amplification. So you can immediately, what you will think of that? You will think of magnesium chloride. That magnesium chloride helps the interaction of the primer with the template DNA. And at higher concentration, this magnesium chloride then provokes the binding of this primer with the template DNA non-specifically. So you will obviously look for reducing the amount of magnesium chloride in the PCR reaction. But if it does not work, then you can think of BSA. I will be telling those additives later on, okay? So I will explain you there. But some of the cheap fixes are that you can pipette out all the 8 ingredients in the bottom of the tube. And what you can do is that instead of putting Taq polymerase in the bottom of the tube, what you do is you put the Taq on the wall of the tube inside, towards the inside cavity. Just pipette out your Taq on the wall of the tube and don't let it mix with the other ingredients. And when you have added everything, you can give a spin to your tube so that Taq can enter into the mixture. And then you can immediately put that tube, PCR tube, in a PCR machine. Okay? Or alternatively, what you can do is you can put the tube in the PCR machine when it is gone to 94 degrees centigrade, add your Taq in there. That is hot start PCR. Okay? That you don't let tag work for polymerization. In the hot start PCR, the tube is at 94 degrees centigrade when you switch on your PCR machine and start the cycles. When the temperature reaches 94, put the tube in there and put a microliter of tag into it, so that there is no chances of tag to start polymerization if the primers have already bound non-specifically. 94 degrees centigrade will again dehybridize primer and the template. And the tag will only start working at annealing temperature. Okay, this is called hot start PCR. You can do hot start PCR, or you can put tag along the wall of the tube, and then you can give it a spin just before you run the PCR machine. Okay. There is a polymerase, tag polymerase, which comes with bound to antibodies. Polymerase is a protein, so they have raised antibodies against this tag polymerase. So what they do is that you mix the polymerase and the antibodies. So antibodies bind the polymerase and don't let that polymerase start polymerization. When you reach 94 degrees centigrade, they are bound in a way, the antibodies and the polymerase, that at 94 degrees centigrade, they dissociate from each other. So the polymerase becomes active again, and it remains active. The antibodies, they get diluted, they cannot bind back to the tag polymerase in the subsequent cycles. So that is one of the methods that you can add. You can start either a hot start PCR, or you can use a tag which is bound to antibodies. In what case? When you are getting non-specific amplification. This is the case for which I am actually talking about. Obviously, you can set up the reaction on ice. You can do physical separation by putting the tag polymerase on the wall, and then all the things are done. You can give it a spin and then put the tube in the PCR machine. And manually delay adding polymerase. That you add polymerase as an 8th ingredient instead of 3rd, 4th, or 2nd ingredient. There is a list of 8 ingredients. So you take your tag polymerase at position number 8, that you add the polymerase as a last step. Make it a habit actually. Make it 7th position, because 8th is water. You can even add it at 8th position after adding that water into it. That is the last ingredient mostly. Okay. So put the pivoting of tag polymerase in a PCR reaction either at position number 7 or at position number 8. So your non-specific amplification will be minimized. Remember that, that's very important point. Touchdown PCR. These names, they are fascinating, but they are very simple actually. Touchdown means, you know, a PCR cycle starts at 94, that is denaturation, then is the annealing, and then is extension, which is at 72 degrees centigrade. A touchdown PCR is that you set the number of cycles to 40. So every cycle has the same denaturation temperature, annealing temperature, and then extension temperature. This is the conventional PCR reaction. A touchdown PCR is that out of 40 cycles, you run the first 10 cycles, for example, where the annealing point is annealing temperature plus 5 degrees. Okay? It means your annealing point is very high. There is very low chances at high bed temperature that the primer will bind to the template, because your annealing temperature is very high. It means annealing point plus 5. If you are setting a PCR reaction at 52 degrees centigrade annealing temperature, set the temperature at 57 for the first 10 cycles for the annealing. And for the rest of the 30 cycles, set the annealing temperature to 52. So initial 10 cycles have a 5-degree higher annealing point. And the rest of the 30 cycles, they have the correct annealing temperature. What it means? That in the first 10 cycles, the amplification will occur only for the highly specific binding of the primer with the template. So obviously, in the first 10 cycles, there will be an amplification for the very specific template. So you will get a lot of template in the first 10 cycles. And then in the rest of the 30 cycles, that amplified template will be available as a template. In this case, the template which is formed at a high temperature is due to highly specific binding of the primer. So you get very specific amplification product in the first 10 cycles. And then you shift down to a lower temperature. Because your template number is very high for the specific one. So your specific template is now being used as template in the 30 cycles. This is called touchdown. Touchdown means run the PCR, the first few cycles, like 5 to 10 cycles at a higher annealing temperature, and then you come down to the calculated annealing point in the rest of the cycles. I hope it is clear. If you have any question, please tell me now. So I will be talking about PCR additives later, so I will not talk about these in this slide. Okay, I have elaborated them more further. Now, there is one more question in our minds, that what are the chances of non-specific binding of a primer? Okay, there is a simple calculation. If you have a 1 nucleotide long primer, 1 nucleotide, it may be either A, G, C, or T. So what are the chances of finding any 4 of these at a very specific point of the DNA sequence? Because there are 4 nucleotides, there is a chance that adenine can come close to G, there is a chance a T can come close to G, there is a chance a C, or a chance a G. So there is 1/4 chance of existence of a C close to G so that they can make a complementary binding between them. 1/4 chance. Now, if you have more number, for example, 2 nucleotides at a position A and G, the chances of finding complementary to an A and G is 1/16 out of 4 nucleotides. Okay. The chances for 3 nucleotides to find the complementarity with the 4 nucleotides is 1/64. If you are looking for a 4 nucleotide long sequence, the chances of finding a complementary sequence to 4 is equal to 1/256. Okay. What it means? It means as the sequence goes longer and longer, the chances to find a random complementarity is decreasing. And our primer in the PCR reaction is 18 nucleotides long. What they have done here is there is a calculation here for a 16 base long, a 16 nucleotide primer. There are chances to bind this primer is 1. There is 1 chance of a 16 nucleotide long primer to bind with 100% complementarity, means from 5 to 3 prime. Is once in 4 billion. If you have 4 billion nucleotides long DNA. How big is human DNA? It's 3.2 billion nucleotides. So a 16 nucleotide primer should have less than one chance to bind to a human DNA if it is not prepared according to a sequence, if it is randomly prepared. Okay. What is non-specific binding? Non-specific binding is that if you decrease the temperature, the primer 3' end binding is very important. Even 3 or 4 nucleotides, if they find some complementarity, we look at 4 nucleotides. There are chances of 100 by 256, okay, which is a very big chance that you will find ATCG at another position, That is the number of times, hundreds of times you will find this sequence, ATCG, on the human genome. If the 3 nucleotides, which are the last 4 nucleotides at your primer will bind at low temperature here, they will do non-specific. So if you have the correct temperature, there is no chance, or 1 in 4 billion nucleotide, that by chance your primer can bind to the big nucleotide sequence somewhere over there. So the specificity is actually very well maintained by the PCR reaction if you just avoid some practices like keeping your PCR reaction at room temperature, okay, or adding polymerase at much earlier stage. And if the GC content in the last 5 nucleotides is less than 2, okay, so just that is called GC clamp. So always see that your primer has at least 2 to 3 G or C's at the 3' end of the primer. Okay, GC clamp is very important. It helps in avoiding the non-specific binding at the 3' end. Okay, so now a few more things about the primer specificity. What is a universal primer? This primer is universal in all the PCR amplifications. Forward is 1, only the reverse is different. So when you have a primer which is universal, which binds to all the related genes, for example, if we talk about ribosomal RNA, you know the whole nucleolus, that's a part of nucleus, has the genes for ribosomal RNA. Okay? So if you design a primer on one ribosomal RNA gene, that primer will be able to bind to all the ribosomal RNA genes which are present in the nucleolus. It means that primer is a universal primer. Okay, it will bind to all the ribosomal genes. If you design a primer against actin, it will bind to all the actin genes present in a cell as homologs, if it has the same complementarity for all the actin genes. So such primers which bind to a range of the genes from the same family, they are known as universal primers. A group specific primer. Now you have a gene family and you have 2 groups. One is acidic protein, one is basic protein. Okay, one gene is coding for acidic protein, the other is coding for basic protein. Obviously, there will be some differences in the nucleotide sequence of the gene. Okay, so if you design a primer on acidic proteins of the same family, that primer will bind to all the genes which are there in that family. On the other hand, the primer that you design for basic proteins that will bind to all that family. So these are now group specific. A specific primer is against only a single gene. It will not bind to any other gene. Okay. So when we talk about primers, primer can be of three different types. It may be a universal primer, it may be a group specific primer, or it is a specific. Okay. So remember in your mind, what is the difference between universal, group specific, and the specific primers? If there is a question, you can ask. So you always used a forward primer and a reverse primer for the amplification of a double-stranded DNA gene or a regulatory sequence or any sequence that on which you have designed the primers. You always need, if you need a double-stranded DNA, you always do use two primers. One is forward, one is reverse. Now the question is, can you perform PCR amplification with a single primer? The answer is not no. It is yes. Okay. Now, there is a technique known as RAPD, randomly amplified DNA polymorphism. In that case, we use single primer. And you already know actually, you are familiar with making probe. Do you use two primers for making the probes or a single primer? You can use one primer or you can use two. What would be the difference? If you perform a PCR reaction with a single primer and if you perform a PCR reaction using two primers, what would be the difference? You have template, you have single primer. The primer will bind to template one strand only, not the second strand. And it will amplify that strand only. So start with second cycle. What you get from a single primer, you will again get 1 copies. So you are not following 2 to raise power n. You are following 1 to raise power n. It's not doubling. One template, 1 copy. Doubling is both of the strands, they make 1 copy. That is doubling. It means if you're using a single primer, the PCR reaction, you are not following exponential growth of the template. That is 2 to raise power n. It is 1 to raise power n. So 40 cycles will not be enough to show you that single-stranded DNA amplification. So using single primer in a PCR reaction, you can never achieve exponential amplification following 2 to raise power n rule. And I hope you understand that rule because I have already told you that. So the last slide of this lecture is that if you face problems in PCR amplification, what may be the reason? Well, there may be many reasons. There might be some inhibitors of PCR. Some of the chemicals in a PCR reaction, if you add them at a higher concentration, they become inhibitors of PCR. Sometimes your DNA can be contaminated with some elements, some compounds, which inhibit the activity of PAC polymerase. Okay? In order to avoid that inhibitors, the best solution is add some BSA, bovine serum albumin. It's a protein. So this acts as a barrier to scavenge the inhibitors. TAC is a TAC polymerase is a protein. Inhibitors are usually protein binding molecules. They bind to the protein and don't let it function. BSA is used at a quite high concentration, that is 0.5 to 0.1 to 0.8 microgram, which is much higher concentration than TAC polymerase. And this protein is highly abundant. It will capture efficiently the inhibitors of the PCR reaction. Okay. So if the problem is with your DNA purity or your chemicals purity, anyway, there are so many things which inhibit a PCR reaction, and you can try bovine serum albumin if you think there is some inhibitor in there. Okay? You have to, this is a hit and trial method actually. You never know in the beginning what is going on. So in one PCR reaction, you add BSA. In another one, you may add DMSO. DMSO, what it does is it opens the secondary structure of DNA, because PCR inhibition can also occur due to secondary structures in the template. And the secondary structures in the template, they are usually due to a high GC content in the template DNA. So DMSO actually helps to remove that secondary structure. Though most common things which you can think of if you have a PCR problem, one is BSA, the other one is DMSO. Glycerol, yes, you can add. This just concentrates the materials. Doesn't have a drastic effect. But try it sometime. If you think that your materials are not assessed properly, or you have lower amounts of nucleotides or magnesium chloride and the lower amount of enzyme, it just concentrates the things. They are very viscous material. Glycerol. Okay. Stringency enhancers. If you feel that you are getting non-specific amplifications, without, by doing, although you are doing a lot of precautions, you know that I have already told you non-specific binding of the primers will result into non-specific amplification. And you want to get rid of that, then you can look at stringency enhancers. They are formamide, detailed, DMAC. Okay? So try a very low concentrations of these materials in your PCR reaction. And most often you will see that formamide is actually the most widely used. So you can have 0.5 microliter of 1 microliter of formamide in your PCR reaction. It is a liquid, okay? Concentrated liquid. 100% absolute. So just take 1 microliter from here, add it to PCR reaction. Formamide. And if you have any non-specific binding of the primer, most of the time you get rid of that. Okay? Non-ionic detergents. Actually, they stabilize TAC polymerase, and they also suppress formation of secondary structure. You can try those, but remember one thing: never ever use STS. STS is a detergent too, but this is non-ionic detergent. STS is negatively charged. It will break down TAC polymerase into its subunits. So never ever use an ionic detergents. And the most important, frequently available ionic detergent in the lab is STS. What others you can use is Triton X100 is always available in some laboratories. Okay, frequently, 220 is available. So you can try half a microliter or 1 microliter of this one. It will stabilize TAC polymerase, and it will also avoid secondary structure. So 9 non-ionic detergents usually, but don't use them in high concentration. Just 0.5 microliter per 25 microliter reaction is enough. Okay.